Hi guys, by now you're on the bus to the Snow River. You've got to enjoy 9 wonderful days on Outward Bound. But as promised, a little podcast to remind you of what we did at the start of the week, because of course mitosis and meiosis and this whole cell replication thing is going to be very important when you come to considering your genetic dilemma. How do we pass on these genes? Why should an achondroplasia parent produce achondroplasia children? And of course, it's all about mitosis and meiosis. So, essentially, there are three reasons why cells copy themselves. If you think back to where we started, cell theory says that all new cells arise from pre-existing cells, and that's for three things. One is this strange idea of making sure that the amount of nucleus within the cell has a certain ratio, has a certain proportion of the cell, and that has to be restored, and cells can't grow beyond that ratio and succeed. So cells are constrained in their size. You guys, of course, are in middle adolescence and you're growing hundreds of thousands of cells every day as you grow into your 10 trillion plus cell adult bodies. So lots of growth and development for you. And of course, the old fellow here with the grey beard is heading in the other direction. And I'm hopefully doing lots of maintenance and repair, and not yet to the point where I'm not replacing all my cells, but still maintaining my body condition. So that's the reason we do cell replication. And you'll find these sort of diagrams around the place. And we talked about it in some detail in class on Monday, how this cell replication cycle takes one cell and creates two essentially cloned copies, so two completely identical copies. And it has a number of stages. So we see G1 as growth, the first growth stage. And I can't point to it. I'll point to it, but on the screen there, on the left-hand side of that diagram is a G1. And at that point in time, the cell is doing all its normal cell stuff. It has, for a human cell, it has 46 chromosomes, and it's starting to put together the materials it needs to copy each of those chromosomes. So then it moves into S stage at the top of the diagram, and in S, it continues to grow. But here it starts to copy all its DNA. So by the end of that blue segment of the diagram, it will have 92 chromosomes in a human cell. Then during G2, the cell continues to grow, and it puts together all the materials it needs to create two cells. So of course cells have mitochondria and Golgi apparatus and endoplasmic reticulum and all those sort of things. And these materials to grow those, it needs to grow two sets because it's going to become two cells. And then we have this stage called mitosis, where those two sets of chromosomes are divided into two new nuclei and two new cells are created. So we have prophase, metaphase, anaphase, telophase. We'll go through those each in a bit more detail. And cytokinesis to finish. And cytokinesis is the rendering of two cells. We've got one cell becomes two, it breaks in half. And that little diagram in the bottom left-hand corner shows those two cells. So, we've done that. We won't worry too much about that one. So here's the interphase. Here's a cell not dividing. It's just putting together all its material. There's a nice big nucleus in the middle. It's probably a young cell from that particular diagram. And it's got a clear nucleus, but we can't really see chromosomes in it. But then we go into mitosis. In the first stage of mitosis, our chromosomes become visible. And here you can see it's not a human cell, it's not 46 chromosomes there, but there's, we've got 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, about 14 pairs of chromosomes there. So it's an animal or a plant. Well, animal, we're not seeing any plant cell walls. Animal cell with 14 sets of chromosomes. And the nuclear membrane's now gone. There's no nucleus, it's dissolved. And we're looking at just the chromosomes. They become quite apparent at prophase, but scattered. At metaphase, they attach themselves to spindle fibers that cross from one pole to the other pole of the cell along the equator of the cell. And so metaphase sees all these pairs of chromosomes lining up, remembering of course that there are two copies of each chromosome. And during anaphase, those copies each go to different poles. Well, that's the plan anyway. We haven't talked about mutation. We'll talk about mutation later on. And there's some problems here that can cause some change and some variation as well. But essentially at this point in time, the one set of chromosomes will go to one end and the other set will go to the other end. We call those the poles of the cell, a bit like our globe, an equator and two poles. And then in telophase, the chromosomes start to complete, the spindle files are gone, and the chromosomes start to coalesce and get back together again to form a nucleus. And in cytokinesis, we can see two nuclei developing, and you can see that lovely new membrane developing between the two cells. And of course, in plant cells, you need to build yourself a new cell wall down the middle too, because two cells will need a cell wall each. And this process is quite, it's coming on in your bodies right now. In fact, you've been working your bodies hard up there in the snowy on your outward bound expedition, so you've probably been growing lots of cells in response to that. And I know both my sons have come back lighter but taller from outward bound, which is an interesting thing, isn't it? But in your body also you have this ability to create cells that have particular roles. So at the top of that diagram you can see what are called embryonic stem cells. So as an embryo you're a couple of layers of a handful of cells. A blastocyst is about 16 cells. This one's a bit more than 16, but it's that early stage. At that point in time, every single gene, and we say it's about 30,000 genes, are switched on. So embryonic stem cells are really interesting to science because all the genes are potentially switched on. But as we age and as that embryo develops into a fetus and into a young human, the cells will become differentiated. They have a particular role to play. And that role is determined by switching on and switching off of the genes that they carry. So as you can see, the nerve cell is quite different to a muscle cell, quite different to a gut cell, different to blood cells. So we have a whole lot of, we have 30 odd different types of cell, and each of them has particular genes that have been switched on, or left switched on, and lots of genes have been switched off. So my classic example I like to talk to students about, of course, is our eye. Our eye is made of cells. They're really, really specialized cells, and they're specialized for collecting light information. So if they decide to switch on the ability to make skin, then my eyes would grow over and I wouldn't see much. I'd actually lose the ability to get light into my eyes and actually use them for finding things. So it's important for those cells to actually talk to each other. We talked about hormones before, and communicate cell to cell to maintain the integrity of that organ, that eye. So we're always saying eye tissue, not other tissues. And some diseases of the eye are caused by the wrong cells growing. And we talked a little bit about cancer in class. Meiosis occurs in our gonads, so ovaries and testes, to make gametes, sperm and eggs. So gonad and gamete are sort of words we use in science to describe, sort of collective terms to describe sex organs and sex cells. So if we think about mitosis, we took one cell and we made two. So we went from 46 chromosomes to 96 chromosomes to two cells with 46 chromosomes. So as you can see in this diagram, reading from the top down, the first stage is identical. So it's mitosis again. And we've ended up with two cells that are essentially identical. Then we go to another second stage of prophase, metaphase, anaphase, telophase. So we do a second mitotic division. We don't copy them again, we just divide them again. And so by doing that, you end up in the bottom corner there, bottom right hand side, with 4 cells. And each of those cells now has one set of chromosomes. So in the case of a human, we're looking at 23 chromosomes. And that's rather handy because we want 23 chromosomes from dad to mix with 23 chromosomes from mum to produce an individual with 46 chromosomes. Because if we made sperm and eggs with 46 chromosomes, we'd have 92 the next generation, 184 the next generation. That number's going to be 360 something, 8 the next generation. And of course, obviously, the numbers are going to get bigger and bigger, and the whole thing becomes unmanageable. So it's really important in sexual reproduction to divide the numbers of chromosomes. So you get one set of chromosomes in each of your gametes. And that's what we're seeing here in meiosis. That's all. Animations I found on the web, and I haven't actually said where I found them from then. I've probably forgotten where I found them now. So apologies to the person I stole that from. This is not mine, obviously. It's off the web somewhere. I don't even know how to do that. And we're seeing here the production of gametes and then the fertilization of one gamete with another gamete and the new individual created. And that one's actually got some deliberate problems in it, but we'll talk about those another day. And it's a second attempt to show the same sort of thing, that migration to poles. And we end up with 4 cells. And there we are. So, why is that important? Well, you guys are all looking at genetic dilemmas, and those genetic dilemmas are inherited conditions. So where do they come from? how do we inherit those conditions? And so, of course, most of these things are found in our genetic material. And then when we make sperm and egg cells, we pass them on in our genetic material. So if I was to develop a skin cancer, I've got this little lump here in my forearm I should have looked at. I won't pass it, well, too old now to pass on to children, I suppose. If I had more children now, I wouldn't pass it on to my children because it's mine. It's my, what we call autosomes. It's in my body cells. But if that cancer was growing in my testes and in the area I actually make sperm, then that cancer could in fact influence the my genes, and maybe mutate my genes, and cause those to be passed on to my next generation. So we're always very interested from a change perspective in what happens in meiosis more than we're worried about mitosis, because mitosis is about building new cells. But if I'm achondroplasic, and a lot of you guys are doing achondroplasia, then every one of my body cells contains the mutation that causes me to be a dwarf. And so every cell has dwarf genes. We'll talk more about that in class as we get our genetic dilemmas underway. Anyway, I hope you have a fantastic time on the Snowy. It's a great camp. You'll work hard, but you should enjoy it and make some really good friends. And I'll see you in about 12 days' time. And then I'll go work out how to stop it.